We're looking at a plant called sticky nightshade or lychee tomato, and that plant is naturally resistant to nematodes, or the pale cyst nematode specifically. And so we're looking to find what gene is responsible for that, and then amplifying that gene and inserting it into potato plants to make potato plants resistant to that nematode. So it's very destructive, and if we found a way to genetically engineer potatoes to be resistant to that, it would be incredible for farmers and crop production. I really wanted to get involved in undergraduate research because I wanted to see if I wanted to pursue research as like a career. And so that was something that really interested me, just getting involved with that. But also I thought that like working with genetic engineering and learning how to do that and new protocols and cloning and things like that sounded very interesting to me. We started off on the computer, so looking at certain genes that we thought could be resistance related, and then we amplified those genes using PCR. And then you mix it with a vector, which is basically a DNA molecule that serves as a vehicle for transferring foreign DNA into another organism. And then we can cut that DNA with restriction enzymes, and since we know where the sites that the restriction enzymes will cut, we can tell what fragment sizes we're looking for. And when we run those samples on a gel after that the plasmid has been digested, then we can pretty solidly determine whether we have the gene we think we're looking for based on the size of the fragments. This experience has been really helpful in making me more independent and more able to look at results and, figure out what they mean for myself, kind of. I think that Dr. Kaplan has been very helpful in teaching me how to interpret gel results, or instruct me on how regular scientists, everyday scientists who do research, conduct experiments or things like that. Getting our genes sequenced and finding out that we even have some genes that were what we thought they were, that's pretty exciting, because we went all the way from computer work of let's pick out some genes and then doing the whole process of amplifying it and cloning it and then isolating the plasmid DNA and cutting it with restriction enzymes. So it's a whole long process, and actually having results is really cool.